Thank you so much for being here for our webinar on the improved detection of low-frequency mutations for both early cancer detection and infectious disease research using our novel, highly accurate sequencing by binding technology. So I'm going to do a couple of quick intros and housekeeping. There we go. So first, to introduce our speakers who are here on the webinar with us. So I'm Alex Sockel. I am the segment lead for cancer genomics at PacBio. And then I'm honored to be joined by two of our collaborators who've really pioneered the use of sequencing by binding for multiple different applications. So first we have Dr. Stephanie Pond, who is the VP of Emerging Opportunities at TGen. She'll be presenting on their work using SBB to detect very rare drug-resistant subpopulations in tuberculosis. And then we also have Dr. Yannis Ragoussis, who's the Professor of Human Genetics and Head of Genome Sciences at McGill University, who will be presenting on their use of SBB for early detection of ovarian and endometrial cancer. And then we also have Dr. Tim Reville, who is the academic associate working with Dr. Ragoussis, who did a lot of the analysis for this work. And so all of us are here on the webinar. Just a couple of quick housekeeping. I'm going to give a quick intro to the SBB technology and how it can be applied, and then we'll go into some pre-recorded talks from our speakers. But as you can see, they are here live with us on the webinar, and so we will be available after the pre-recorded talks for some Q&A. So then just to quickly orient you to the GoToWebinar actual screen here, all of you are muted just for clarity. Also, we're recording the session, and so in about a week, we will send out a link to that recording. If you have questions during the presentations, if you look up to the top right of your screen, there is a little question mark tab where you can actually enter questions during the presentations, and then we'll be able to answer those at the end. Also, right next to that question tab, the little icon with the paperclip, we also have uploaded some complimentary literature there, so please feel free to download that and take it with you. And then finally, there's going to be a 2-minute survey at the end of this webinar, and we really hope that you will take the time to fill it out, as it really helps us do a better job of planning these webinars in the future. So with that, I am going to go ahead and get started, and we're going to go off camera. Awesome. So I'm going to give you, as I said, a short intro to sequencing by binding, which is the novel chemistry that gives the ONSO system its remarkable accuracy. There we go. So first, why do we care about accuracy in sequencing in the first place? I'm going to use an example here from oncology, which is one of the applications that you'll hear about today. And so to the right here, we're showing ctDNA levels for a typical cancer patient, shown in blue. And so with current approaches for liquid biopsy, which is what we're showing here, the sensitivity for ctDNA detection is limited by sequencing errors. So these increase the noise and obscure the signal, and that therefore limits the detection at which you are able to detect the ctDNA. So in contrast, ONSO is 10 to 100 times more accurate than these existing methods. So this means you can have fewer false positives, so less noise, which then lowers the limit of detection and potentially enables earlier detection and improved monitoring of cancer. So in practical terms, what this means is the greater accuracy allows you to push the limits of detection beyond what you currently think is possible to achieve even greater sensitivity. By reducing coverage requirements, as you'll see in a minute, it means you can sequence less to achieve similar results, which then lowers your cost. It means you can sequence complex regions of the genome like microsatellites with greater resolution than ever before. And our accuracy means that you can be more confident that what you see is real. So how do we actually enable this remarkable increase in accuracy? It is all in our chemistry, which again, we term sequencing by binding, as opposed to sequencing by synthesis, which is what most existing methods use and what I am showing here on the left. So to quickly give you an overview here, in SBS, you start with an unblocked 3' end, and then you flow in blocked fluorescently labeled nucleotides, which bind and are imaged, and then you chop off the fluorophore and remove the block all in one step to then incorporate that base. The problem with this approach is that what you are left with is something we term molecular scarring. So essentially the linker arm that was attached to the fluorophore is left on the incorporated nucleotide. And this results again in molecular scarring, which the polymerase doesn't like. So essentially what this can result in is misincorporation events, so the next base being less likely to be accurate, as well as premature termination of the extension. So these are some of the challenges with existing methods that lead to sequencing errors. Sequencing by binding, in contrast, separates the interrogation and incorporation steps. So here what you can see, we start with a blocked 3' end, and then you flow in labeled nucleotides which bind transiently and are imaged, and then we do a very stringent wash to remove these labeled nucleotides, and then we remove the block. Then we flow in unlabeled nucleotides, and it's those unlabeled nucleotides which actually get incorporated into the growing DNA strand, leading to a more natural extension product and therefore a happier polymerase, which then can do a better job of incorporating the next base. To give you a sense of how much more accurate we're actually talking here, I'm showing a plot with the mean Q scores per base. So you have read 1 and read 2 here. And we have ONSO shown in pink. We have elements of VD cloud break technology shown in purple, and then we have NovaSeq 6000 shown in orange. And so just to orient you, this is from a PCR-free run with HG002, so the data for Element and NovaSeq was just downloaded straight from their websites. And so the first thing you can see here, our spec for ONSO is 90% of bases greater than Q40. But here what you can see is that actually the ONSO's Q scores are above Q50 for the vast majority of the read. This comes out to an average error probability of around 1 in 270,000. And it also doesn't show the characteristic drop in quality towards the end of the reads, which is the result of accumulated errors over time. So again, in contrast to these other technologies, we have significantly greater Q scores. So here we had 100% of bases were above Q40 and 85% of bases were actually above Q50. So significant improvement over existing technologies. Beyond just the straight accuracy, I'd also like to go through a few of the other advantages of SBB over SBS. So the first here is our ability to sequence through challenging regions like homopolymers. So we mentioned this very early on, and that's what we're showing in this IGV plot on the right. So you'll hear more about this in Giannis's talk in just a bit, but this, there we go. More time, there we go. The ability to sequence through these complex regions is really important for things like the detection of microsatellite instability in cancer, since the vast majority of microsatellites in the human genome are actually AT homopolymers. So here what we're showing in the IGV is actually one of those regions. This is a poly A repeat, which you can see here in the center of this region. And with SBS on the top here, what you are seeing is the results of a known issue called post-homopolymer errors, where the polymerase just keeps adding the same base even after the repeat has ended. And so this results in the accumulation of errors on either side of the repeat, as you can see here shown with the colored bases that don't match the reference, and an inability to detect small changes in the repeat length, as you can see. So here, in contrast, SBB is exceptionally good at sequencing through these homopolymer regions. And so here you can see that we are very clearly able to resolve this single base pair deletion in that repetitive region. Some other advantages of SBB are listed over on the left here. So in particular, reductions in index hopping and duplication rates, both of which reduce the amount of redundant or wasted sequencing in your run. And these advantages result in part from the way that we do clustering, which uses rolling circle amplification rather than exponential or bridge amplification. So not only does this increase the accuracy by always copying from the original template, so this prevents any errors introduced in an early cycle of clustering from propagating to the entire cluster, but it also reduces index hopping, since again, a single misincorporation of a leftover primer that's containing the wrong index, it won't get propagated throughout the entire cluster. And again, because with rolling circle amplification, you are making one big molecule with all the copies of your template attached to each other. You also reduce your duplication rates because you don't have template molecules that float away to seed an adjacent cluster. And finally, ONSO also requires less input going onto the sequencer, which allows you to do fewer cycles of PCR, again, reducing sequencing error or PCR-based errors and duplicates. And then again, as I'll show in a minute, you can also achieve the same results with less sequencing. So this reduces your cost per sample. So really a huge array of advantages here with SBB, from accuracy and really beyond. So that all sounds great, but let's see the data. How does SBB actually perform for something like liquid biopsy, where you're trying to find a needle in a haystack? So here, the experiment that I'll show you results from was based on a cfDNA reference sample, which has variants at known allele frequencies. And we did a head-to-head comparison of SBB versus SBS using a third-party library prep and capture kit, and then our ONSO conversion kit to convert the P5, P7 libraries to be ONSO compatible. And really, we did this to minimize any variability that's caused by stochastic sampling of low frequency alleles if we were to do multiple library prep. So here we did library prep, capture, and we split the post capture into 2 aliquots, one of which goes to SBS, the other gets converted and then run on SBB. So first, taking a look at a single variant site within that reference sample, here we're looking at the IGV for the NRAS Q61R variant at 0.1% frequency in the SBS data. And so here, if you look in the zoom in window here, as well as the counts, the base counts in the table, what you can see is that we are actually with SBS getting base counts for all 3 alternate alleles here, which makes it completely unclear which of these is actually the true mutation, or indeed if there is any mutation present here at all. It's possible all of this is sequencing error. And so again, if this were not a reference sample, we would have no way of knowing which of these alleles is the correct mutation. So then if you look at that same site in the SBB data, you can see that the IGV is completely clean with no noise due to sequencing errors. And then if you look at the base counts in the table, indeed, we are only getting calls for 1 alternate allele, which is the C in this case, making it really obvious what is the true variant. So that was for a single variant. What does this look like across all variants? So here what I'm showing you is sensitivity on the y-axis. So that's the percent of the known variants that were detected by each technology. And here we're looking at the 0% allele frequency, which is the wild type, the 0.05% allele frequency, and the 0.1% allele frequency. And here what we're showing is the apples to apples comparison of 12,000x SBB with duplex UMI correction compared to 12,000x SBS also with duplex UMI correction. And so here what you can see is in this apples to apples comparison, SBB is doing significantly better at detecting these low frequency alleles, both at 0.05% and particularly at 0.1%, where you can see the sensitivity is up above 90%. So achieving significantly better detection of these low frequency variants. And then here what we're showing is essentially the same plot, but this time what we are comparing is 6,000x SBB with no error correction at all to 24,000x SBS, again with duplex UMI correction. And here what you can see is that even with 4-fold less sequencing and without doing any sort of error correction, SBB is able to do approximately equivalently in terms of its ability to detect these low frequency variants. So you can sequence the same amount and achieve greater sensitivity, or sequence dramatically less to achieve the same level of detection ability. And so to dig into what is actually causing this increase in recall, here I'm showing a plot looking at the estimated or observed allele frequency for variants in the samples. This is that 0.1% allele frequency sample at 12,000x, or the wild type, so any variant you detect in the wild type is a false positive, compared to the 0.1% sample. And so here what you see is across 10 different subsampling replicates to 12,000x, we observed a total of 2 false positives in the wild type. And the allele frequency of those false positives is actually below the allele frequency of any of the variants that were called in the sample. And so what that means is if in the context of a clinical test, you are trying to apply a very high specificity threshold, you would still be able to call all of these variants confidently because the frequency of false positives is below the frequency of these variants. So that's for SBB. In contrast, if you look at the same data for SBS here, shown in orange, you can see that there are a ton of false positives in the wild type, and the allele frequency of these false positives actually significantly overlaps the allele frequency of the variant seen in the true sample. And so what that means is, again, if you were to set this very high specificity threshold as required for a clinical test to reduce false positives, you would not be able to call any of the variants that are here below that threshold. And so really then any mutation here which may have relevance, biological relevance for an individual is really not going to be able to be called. And so really what we are seeing in terms of this increase in sensitivity is actually driven by the reduction in false positives, which allows us to set that limit of detection much lower for SBB compared to SBS. And so while Onso's accuracy is obviously awesome for this type of low frequency variant detection, it also gives you the highest quality data across applications. So spanning WGS to single cell RNA-seq to wastewater profiling. We actually had some really interesting data on wastewater profiling recently, methylation detection, really across applications. Onso is going to give you the highest quality data and the greatest accuracy for the greatest confidence, no matter what you are doing. And just a final point here, Onso is our mid-throughput platform, which is shipping now, but we've also announced that we are in the process of developing a high-throughput SBB instrument, which is going to have output on the order of a NovaSeq X or X Plus with that same high accuracy of the SBB chemistry. And what we're really excited about is with our recent acquisition of Apton last year, it actually allowed us to speed up this development process dramatically. So stay tuned. We are excited to get this out there and into customers' hands. Thanks so much. And now I'm going to turn it over to the talks from our speakers.
Good morning, everybody. Thank you so much for the opportunity to come and talk to you about some of the work that we've been doing at TGen using our Onso system to look for rare variant detection. So TGen is a nonprofit research institution that focuses on a number of different application areas. And my team really evaluates new technologies to come into the organization for multiple application purposes. So why were we interested in looking at the Onso for ultra-high accuracy short reads? So we want to detect populations that are associated with disease in a high background of non-disease material from small clinical samples. And two applications that I'm going to talk about today are in oncology and also in infectious diseases, where in both applications, early detection really improves patient outcomes. And to do earlier detection, we need more and more sensitive tests, which means detecting a smaller and smaller population. And in a technology framework, that translates to having being able to detect variants of lower and lower frequency. Now, we're all very familiar with doing this in SBS or SBS chemistry, which is what Illumina uses, and a whole host of methods around error correction to drive the limit of detection down have been published on. UMIs are probably the most popular, but duplex sequencing as well as other methods are all used with the primary goal to drive down the error rate that's detected in sequencing to allow us to report on variants of lower and lower frequency. And these methods are incredibly effective, but they all add complexity and additional cost to the assay. So we wanted to look at how we can use PacBio sequencing by binding technology to identify these rare populations without error correction methods. And the first project I'm going to talk about is really detection of drug-resistant tuberculosis. And this work was all done through early access with PacBio on a pre-commercial prototype of Onso. And so that data is now a little bit older, but I think it really shows the power of the system. And then I want to talk about the newer projects that we're doing in oncology on our commercial Onso system that we have at TGen. So first, I want to talk about our project in drug-resistant tuberculosis. And this is really an important area to study because tuberculosis is still a leading cause of death worldwide. It's one of the most common infectious diseases. And over 4,000 people a day still die from tuberculosis. It's not just a disease that occurs in other places in the world. 5 to 10% of Americans test positive for TB. And what's really critical in driving this disease is that up to 20% of infections are estimated to have some form of drug resistance. And the percentage of multi-drug resistant and extensively drug resistant strains is rapidly increasing, and those are harder and harder to treat. Antibiotic resistance really starts where you have a small population of your pathogen that has a specific variant that occurs because of the therapy that's happening. When you treat this disease, the antibiotics kill all of the population that doesn't have that variation, and then that antibiotic-resistant bacteria grows and takes over. And so identifying those rare populations early before the therapy begins is really important so that these antibiotic-resistant populations don't become dominant. Tuberculosis also happens to be an outstanding model system when in early development of a technology like the pre-commercial version of the onso. It's a 4.4 megabase genome that has 4,000 genes. It's challenging because it has a high percent GC, but it also has known hotspot genes for antibiotic resistance. And so our study really focused in on two genes, GRA and CATG, that are really the two genes that have the strongest drivers towards extreme drug resistance. Our experimental design was pretty simple. We took known well-characterized plasmids for either mutant or wild type mutations, and we amplified out the gene of interest. This little red line indicates conceptually that where the variant is. So we PCR that out. We do a second round of PCR to add on our sequencing adapters. And then we take that product and we make mixtures of different levels of mutation versus wild type. We then took those and did 3 different experiments. We split that in half and we sequenced half of it on an Illumina MiSeq SBS chemistry system. And on that Illumina data, we did 2 different analysis types. We looked at just single read data, uncorrected, and we also used a duplex approach to impose error correction on the system to look at what the error corrected data looked like. We took also aliquots of this material and we did 5 cycles of additional PCR to convert those into the SBS PacBio system. And we sequenced those on their pre-commercial system where we have single end reads. And that is a function of where the technology was at the time that we did this study, although now they have the capacity to do paired end reads. To show the impact of high accuracy reads that the SBB chemistry produces, we've plotted here the raw reads from both the MiSeq as well as the SBB chemistry in terms of raw reads in IGV. The top panel is data off of the MiSeq, and the bottom panel is data off of the PacBio SBB sequencer. And intuitively, you can immediately see that the reads are much higher accuracy. The colored dots that you see on the top panel are the errors or the errors in the Illumina sequencing data compared to the plasmid reference. And so you can see that there are a number of errors in the MiSeq data and many fewer errors in the SBB data. Here you can see the data where we spiked different levels of the variant into a background of wild type. And along the x-axis, you can see the expected variant frequency. Along the y-axis, you can detect the observed frequency. And you can see on the panel on the left for CATG that we titrated down this very nice linear titration curve, and that we were able to detect the mutation that we were looking for down to 0.001 above where the wild type reads were. The performance in the other gene, GRA, wasn't quite as good. We had a higher background in the wild type, and so we were able to detect this variant down to 0.01%. Now, this is done at 100,000 read depth, which isn't really statistically significant for variant calling, but because we're looking for a single known variant, I think it really highlights the potential power of SBB chemistry. If we were to downsample the data to 20,000 read depth, which is really what we would do operationally in the lab in a tuberculosis typing experiment, we here show the comparison of the 3 different methods. So we have 20,000 read depth for, here I'm showing the data for CATG, and you can see on the left panel, we have SBS. So this is the uncorrected SBS data. And you can just see that this titration curve is linear and has good confidence, down to the 0.1 to 1% level, which is consistent with all of our expectations around uncorrected Illumina data on a MiSeq. The middle panel shows what that error correction looks like using SBS. And because we're looking at a single point here, it looks very similar to the unerror corrected data. The SBB data, on the other hand, which is shown in the right panel, you can see that the confidence interval in the linear detection range extends to 0.1 to 0.001%. So a full order of magnitude lower than what we see with the SBS data. If we zoom out now and stop looking at the thing that we know is there and instead look across the entire gene, you can see that we've calculated the error rates across the amplicon. And I really want to highlight that this is observed error and not a true error rate, because in this experiment, we have not separated out sequencing error from the PCR error that would occur as an example. And additionally, the SBB data has gone through an additional 5 cycles of PCR. So this is really observed error rate and not a true sequencing error rate. But what you can see is that for both genes, the error rate for the SBS data is plotted in orange, and the SBB data and the error-corrected SBB data, which is labeled here as SBS SMORE, is around 8 times lower than that of what you see in uncorrected SBS data across the entire amplicon. So we really do see across the entire gene this impact of high accuracy reads in this experiment. Now, accuracy really translates to a reduction in false positives. And we all know that this is really one of the limitations when you do variant calling, is that you can't distinguish between a true and a false positive. And so that sets your threshold for what you report on. And if you look at the 3 different method comparisons, you can see what you expect when you look at variants that are occurring in the 1 to 10% range, right? You see a few false positives in this range from SBS, and you see almost none in the error. You see none in the error corrected in SBB data. If you go down to the next row and you look at variants that are occurring between 0.1 and 1%, you can see that we're seeing a lot of false positives with the SBS data, 168, and a significant reduction in both the error-corrected data and the SBS data. And you can see that as you go to lower and lower thresholds, you see more and more false positives, but the SBB data is consistently lower than the other methods. And this should tie back to that IVG plot that I showed earlier, and that intuitive view that there's fewer variants translates here into the quantitation. So we were quite happy with the performance in this pre-commercial version of the SBB chemistry, and we saw about 8 to 10% improvement in error rate. And so we went ahead and purchased the commercial ONSO system, and we're now using that to explore different applications in oncology. And specifically, we're interested in using ONSO for liquid biopsy. And I'm sure you're all very familiar with this technology, but it's a blood test that allows you to look for highly fragmented cell-free DNA to detect disease. So as our first project in this space, we're sequencing 250 plasma clinical specimens. Half are cancer, half are normal. The cfDNA gets isolated out of the plasma. It all goes through the Watchmaker Genomics Standard Library Prep protocol. And after it goes through this protocol, it gets split. Half of the sample gets sequenced on a NovaSeq 6000. Half of it gets sequenced, half of it gets converted into an ONSO library. So again, that's at 5 additional cycles of PCR. And then it gets sequenced on the PacBio ONSO. And the reason that we're doing this conversion instead of a native library prep is just, as you all are familiar with, you get so little DNA from clinical plasma that splitting it was the most viable option for the experiment. So the first question when you try a new technology is how fast can you convert over to the technology? So here I have a burn down chart that really shows how fast you can do this. So the red line is really that PCR conversion step to make libraries. And you can see we did that very, very quickly. Is this nice baseline here because we had some really low quality samples that we weren't sure if we were going to be able to sequence those. And so we just sort of stuck around and waited. In terms of sequencing, we expected the lab to run two flow cells a week, and that's shown here in the yellow curve. And then the green curve shows actually how many flow cells we were able to sequence a week. So you can see that once we started the project and had the instrument installed, we were pretty rapidly doing 2 to 3 flow cells a week. Once we got through most of the samples, we really slowed down. And the reason that we did that was because of these low quality samples where the Illumina sequencing data didn't come out particularly beautifully. But we decided to give them a try. But we did sort of this by category where we went from high quality samples and then in the next flow cell, slightly worse quality, worse quality, worse quality. And so this was really slowed down because we were doing this flow cell by flow cell and then assessing to see how the instrument was performing with that category of quality. So I want to show you some of the data. So here's a whole series of flow cells that we've been running and their cluster density. You can see that it's fairly consistent. In the very early sets of flow cells, we were still titrating it in. And so we would try to increase if we'd go a little high, adjust and go a little bit low. But overall, it's been very consistent. And I want to remind you that some of these last flow cells were with samples that we weren't sure we were going to be able to sequence. They had really high primer dimer, but we were able to get the data. So we also, here we're showing the projected yields. You can see that that's very consistent. We also look at a metric percent undetermined, and you can see that's fairly high, although within the range that PacBio indicated that we should expect to see. And we talked to them a little bit about this. And percent undetermined is really coming down to we're seeing some issues in demultiplexing the converted libraries. And they're seeing this value to be much, much lower on the native libraries. So our hypothesis moving forward is that there's probably some indexes that are in the Illumina panels that don't necessarily translate as well to this new chemistry. I think that we've all seen that before in switching instruments, that there's just some indexes that don't play as nicely on certain instruments. And so this is something that we're looking to dig into a little bit more, but it hasn't slowed us down. You can see the read totals that we're getting. And most importantly is this bottom graph with the metric of around base quality. So this means that 90% of the bases have this particular quality metric, quality Q score or higher. So this is the minimum quality in 90% of the bases. And you can see that across all the flow cells, we're getting that to be around 40 Q40 or higher. The single exception is this flow cell 3, and that's the one where we over clustered it as we were trying to drive the cluster density up. Here you can see for the first 20 samples, the average Q score per cycle. And so you can see on the ONSO system, we're regularly getting quality scores of Q50 or higher. And you can see what those equivalents are on the SBS sequencer on the NovaSeq 6000 are around 35. So we're getting excellent data quality. A few other things that I thought you would find that are interesting is this metric around the average reads per sample, because many of us have really seen over the years that on Illumina sequencers, there's a really high variability in how samples, the number of reads per sample when you multiplex them and sequence and then demultiplex them. And what we're seeing on the ONSO is it is much more even sample to sample and run to run and much more predictable. And we're not seeing the variability that for this sample set that we see on the SBS sequencer. we're seeing very high passing filter rates for the PacBio onso, as shown in pink. We've had kind of a drop off here on this most recent flow cell, but again, we're now down sort of into those samples that we didn't think we were going to be able to put on this sequencer that have incredibly high percent primer dimer and didn't perform particularly well in other experiments. The other really interesting metric that was kind of a surprise to us is this percent duplication rate. And so you can see that the PacBio onso has a much lower duplication rate than the SBS. And this really surprised us because these onso samples went through even more PCR than the SBS did. And so we've reached out and PacBio's confirmed that they're seeing this consistently, not just across our projects, but others as well. And so I think that was a little bit of a surprise, and it's a potential real benefit because we are able to use more of the data. So that's something that we're tracking very closely to understand what's driving that. So this data set has been fully generated now for all 250 samples. And so now we're really digging into the data analysis to understand, to really do that analysis around our ability to distinguish cancer versus normal samples in our liquid biopsy projects. So hopefully we'll be able to present on that data in the fall. And we're really excited to share that with you. I want to thank everybody who contributed to this project, especially those at PacBio who helped with the pre-commercial early access for the tuberculosis project, as well as getting our onso system set up. And the really fantastic group from multiple research collaborators at TGen who've contributed samples and data analysis to understanding how this instrument works. Thank you so much for your attention. I hope I was able to share with you how the onso is performing in our applications, and we think it's really promising, and we're very excited to continue to leverage it in our research programs. So thank you.
Thank you for the invitation to present in this webinar session. I'm Yanis Dragousis, professor in human genetics and head of genome sciences at the McGill Genome Center. The work I'm going to present is about highly accurate sequencing by binding for improved detection of low frequency mutations, and it's a collaboration with my academic associate, Tim Reville. The project on which the data I'm going to present is based is about the early detection of gynecological cancers using a molecular genomic assay, whereby, as you can see here on the left, a sample is being taken from the inside the uterus of the patient, as well as a saliva sample. We perform DNA extraction, followed by panel sequencing, which is based on a capture using Agilent SureSelect technology in this case, and high sensitivity sequencing, aiming to detect one germline mutations in order to identify the possibility of an underlying hereditary risk for these cancers, and also somatic mutation detection, whereby we have developed a machine learning based algorithm that gives us a classifier based on the individual patient data that categorizes the results into either positive, indicating the possible occurrence of an ovarian, fallopian tube, or uterus tumor, or a negative result for the same types of tumors. And so far we have processed over 5,000 samples in this work. How we perform the high sensitivity sequencing, this is based on duplex sequencing technology, which is established a few years back, and we are following it through for the duration of this project, which is ongoing for over 3 years now. And the duplex sequencing technology, as you can see here, relies on the following principle, that you generate from the DNA, you generate short read sequencing libraries that are utilizing adapters with a specific pattern of unique molecular identifiers, which allows one to actually reconstruct the original DNA fragment from the sequences that are obtained and identify the top and the bottom strand of the original DNA fragment. I will explain this a little bit more. So after capture of these fragments using Agilent technology, here on the right, we amplify by PCR in order to generate duplicates that come from the top strand as well as from the bottom strand. These are used to generate a consensus, which is put together by reading the unique molecular identifiers that belong to an original DNA fragment. And then the top and bottom strand data are merged to reconstruct the sequence of the original fragment and identify the possible presence of true mutations. And we have based this on a commercially available kit, which is based on SureSelect capture technology that incorporates this type of unique molecular identifiers in the form of the SureSelect HS assay. So for this assay, we use a 32 gene panel. For most of the genes in this panel that are relevant for ovarian endometrial cancers, we capture all exons. We also capture some established hotspots for the occurrence of somatic mutations in these cancers. We also capture 48 genotyping SNPs and microsatellites. I'm going to describe this towards the end of the presentation. The total capture length is region is 134 kilobases, and we aim to get a coverage of at least 1,000 duplex read, and for that we need a raw read depth of over 10,000. So we aim for 22,000 fold coverage in terms of raw reads, which corresponds to about 20 million reads per sample that we are generating. So it's quite a lot of reads per sample. And now for this presentation, we are testing the performance of sequencing by binding in duplex sequencing, and we ask some key questions like, can the ONSO sequencing by binding platform improve the performance of duplex sequencing by reducing sequencing errors? We want to evaluate the potential of the sequencing method to detect low-frequency mutations with no need of duplex sequencing, and also to test the potential of sequencing by binding to produce higher quality reads over difficult to sequence regions as applicable in the detection of microsatellite instability at high sensitivity, for example. So in order to do that, we took 39 samples derived from saliva and brush from both benign conditions, endometrial and ovarian cancers. These were captured using the SureSelect HS from Agilent and sequenced in parallel, on the one hand in Illumina NovaSeq 6000 instrument, but as well as in the PacBio ONSO, following a conversion of the original Illumina libraries into the PacBio libraries. And again, aiming for 20 million reads per sample as usual. Initial data quality controls and some comparisons can be shown here. where we see that actually, indeed, sequencing using the PacBio platform produces very high Phred scores. As you can see here, at between 52 and 54 for read 1 versus the Illumina standard for the standard in terms of performance of our system at 36 to 38 Phred scores, which is quite good. But obviously the PacBio also is higher. However, does this translate to any tangible benefits? So here we can see the match rates of the raw reads here on the left in terms of the Illumina platform, sequenced in the Illumina platform versus in the PacBio platform. And you can see here already that these are higher in the PacBio raw data, going through to the different iterations of including duplex sequences. And here we can see the raw duplex sequences, whereby in pink, the PacBio data perform very well, but also obviously also the Illumina data once we reach the duplex sequencing stage. Inversely, the mismatch rates are lower in the PacBio, as well as overall the deletion and insertion rates that are lower, but particularly in the raw reads. Okay, that's good, but does this translate to a tangible benefit in duplex sequencing? Indeed, when we look at the performance of the PacBio data on the left versus Illumina data from the same libraries, what we can see is that actually, as you can see here, the PacBio data gave us a high percentage of duplex reads out of the same number of raw reads. So we can see already a positive effect of the high accuracy and match rates, if you want, of the PacBio data. Now, we investigate, does this influence actually the somatic variants that we can call from this data? In order to be conservative after duplex deduplication, calling of variants using VARDICT, we set a relative stringent criterion of 5 alternate reads. So 5, and what I mean by that is 5 DNA fragments producing the same call, and we wanted the regions to be called at 1,000 fragments at least in both platforms, so that we can compare them, and we remove variants that involve more than 50 base pairs. So we intersect and see what happens. What we can see immediately, and this is another benefit of having higher accuracy reads, we can see that the number of bases that were callable were much higher in the PacBio platform versus the Illumina platform. So we get more regions that we can score with 1,000 fragments or higher when we sequenced with the also SBB. However, we look here only at the intersection. And what we can see is that it's very good under these criteria. There is very good agreement between the two platforms. So both give us more or less the same calls, with the only difference that Illumina gave us, the Illumina sequencing gave us a few more calls. So then we asked ourselves, what are these calls, and what is their extra really in the regions in which the calls have been made? And what we can see immediately is actually that the if we look at the sequence context within, left and right of the sequence that have been called as containing a variant, you can see with Illumina that these were regions that had, at very low frequency, they had mismatches, which are not observed in PacBio. To cut the long story short, this was seen in the other extra calls made using the Illumina sequences. So then we can ask, can the sequencing errors propagate in duplex reads? And yes, and this happens when we sequence the top, they are double-stranded fragments derived from the top on the left or the bottom strand. They are sequenced using classical paired-end sequencing, read 1 and read 2. But the bottom line is that when we sequence with either read 1 or read 2, both the top and the bottom strand are sequenced in the same orientation. So, for example, if sequencing the bottom strand, for example, it has a problem in the reverse. The top strand has a problem sequencing in the reverse orientation, then this can happen very well also in the corresponding bottom fragment. And if this error is systematic, then it will propagate in our data and produce an output that would indicate the presence of a mutation in this particular position. And here we can see it. We have the two top and bottom strand, and we look at reads that have been generated in the reverse orientation of the original fragment, and you can see here marked in red for the Illumina sequencing, it produces low quality reads over the same part of the sequence. These are low frequency, however, they are very low quality. In contrast, the PacBio ONSO data are systematically clean and do not produce any extra calls over exactly the same region. So these can be observed only in the Illumina calls. And this is visualized in IGV here. At the bottom, these are the raw reads, and you can see that the Illumina raw reads do contain many mismatches to the original sequence over the same region. The PacBio data do not. And then if you use duplex sequencing, these become absolutely clean, while in the Illumina, these errors, many of these errors are propagated. We have seen this also in germline calls, so where we compare 30x coverage, and again, you use conservative calls. There are some regions that produce differences in terms of the calls between PacBio and Illumina, and you can see here in the bottom, these are Illumina sequences. So there are many bases that are called different from the reference, with some insertions shown here that are called. In PacBio data, we get only one type of call, which is an insertion, and the call is a CTT insertion versus a CTTT shown at low frequency here in the Illumina data. But you will have low confidence due to the presence of the extra calls in mismatches in this region. So we also compared whether we can detect low frequency mutations without the need for unique molecular identifiers. So as you can see here, we use the Illumina data to generate duplex sequencing data to identify mutations. In this case, we use 12.8 million raw reads to produce duplex sequencing and score somatic variants in the frequency spectrum that it would be interesting to investigate, mainly between 1% and 0.1%. And what we can say so far is that we can detect these mutations also with the PacBio ONSO data without non-UMI deduplication and by utilizing 5 million raw reads. So, but as I'm going to say, there is more work to be done to take advantage of the high quality of the data to produce calls at very low frequencies. Now, finally, for the high sensitivity detection of microsatellite instability, this is very important because detection of high microsatellite instability or mismatch repair defects is applied, in Lynch syndrome cases and also in endometrial cancer, and is important in other cancers, but it's important for therapy decisions because immunotherapy could apply in these cases. And here we ask the question, if we use our brush samples, can we detect microsatellite instability? To answer this question, we also use patients from which, samples from which derived from the tumor were used to detect by other methods microsatellite instability and controls. So we took our data and processed them with MSI sensor. And what we can see is that indeed, shown here, we can detect microsatellite instability. We can detect it with both platforms, whereby in order to get good coverage, we use hybrid data. Again, in many cases, the sequences derived from the PacBio Onso detect more positions with microsatellite instability overall. And as you can see here, the other samples are negative. So it works quite well and it can be visualized again. So this is now raw data over the microsatellite region. At the bottom, you can see the Illumina data. It's difficult. You get a lot of mismatches close to the sequence of the microsatellite, and the deletion scored, for example, have an extent that can fluctuate a lot from one read group to the next. With the PacBio data, you can see immediately that the detection of the instability is very sharp and quite consistent between the different reads. So we get better quality data, which can be very important, particularly when we don't have a lot of coverage, as shown here in some regions. So to finalize, the sequencing by binding is a promising new technology. It produces superior sequence read quality than the current Illumina standard, leading to improvements in read mapping quality, is suitable for duplex sequencing application. The read quality leads to improvements in raw read utilization for duplex sequencing applications. Sequencing errors can affect duplex sequencing outputs, as we have shown in some cases, and the Onso platform reduces their occurrence, reducing spurious results. It performs well over challenging regions, such as the microsatellite sequences, and is suitable for sensitive microsatellite instability detection. Therefore, it gives us the possibility to increase the sensitivity and accuracy of our assay with this new sequencing by binding technology. And another conclusion is that specific analysis tools need to be developed that utilize the increased accuracy of the platform to detect low frequency variants in particular. I finish by thanking my group, and particularly Tim Reville, who did the analysis I presented for this work, and the rest of my team for producing the data, as well as the clinical team of Professor Lucy Gilbert, who leads this project and informed particularly of a clinical trial that is ongoing. Many thanks. And if you have any questions, I'm happy to answer.
Awesome. Thanks everybody for the claps. It's really nice. We super appreciate you guys staying on. I know we're a little bit over time. We do have a couple of questions in the chat that I do want to make sure that we address. So if you're able to stick around for the Q&A, we would appreciate it. So the first one, this is more about the specifics of the onso sequencing chemistry. I have a question about the chemistry of sequencing by binding. What is the length of each read, and is it paired ended? So I can take that one. I think Stephanie did touch on this briefly in her presentation, but the chemistry that Stephanie used in the project in the very early days of onso was single-ended, but we now do have paired end chemistry. So we have two kits. We have a 200 cycle kit and a 300 cycle kit. So you can do 2x150, 2x100, 1x200, a lot of different read configurations. So we do have that available now. Another question, I think, for Giannis: how does SureSelect identify SNPs from one copy of the chromosome only at 50% frequency? I'm not totally sure. I think just more question on how SureSelect actually identifies SNPs.
Yes, so it's a capture-based method. So it does identify SNPs. It depends on the design of the assay, but even if you go whole exome, it will identify SNPs that are associated with an exon or flanking an exon. So it depends. In our design, we have included intronic sequences, flanking intronic sequences as well. So it will identify SNPs. It's a matter of design, basically.
Awesome, thank you. Question for Stephanie. In the tuberculosis work, have you tried using error correction with onso, or what do you think the impact would be?
That is a great question and something that we're very interested in trying, but we haven't yet. I think we'll probably do that on a different test system and not tuberculosis, because in the tuberculosis applications, because there's so many PCR cycles to amplify out the pathogen of interest, I think what we'll see is actually a measurement of the polymerase error rate. And so at some point, we're going to hit the floor on error correction. So I think there's more interest at TGen in looking at combining those error correction methods for oncology on the onso. And yes, it is definitely on our list of experiments.
Awesome, thank you. This is probably really for both of you, also related to error correction. Do you see the advantage of accuracy as really replacing error correction methods, or is it more complementing or improving them? Either of you go.
Stephanie, do you want to start?
Please go right ahead. I'll follow on.
All right. Thank you for the question. So I can see as doing both. Basically, it can push the limit of sensitivity and accuracy without using any type of tricks with UMIs and duplex sequencing and other methods. I would say that it's also important to remember that some errors do occur through the library prep process, and I think if we want to go to achieve very high levels of sensitivity, it's important to address these, because these are not sequencing errors anymore. They are library prep errors, so we should have this in mind. But I can see, at least from our experience, that it can do both. It can improve even further what can be detected through error correction methods. So it can do both.
I agree completely. We'll look at both it depends on your application. I think I'm really excited to start putting PCR-free libraries, or as close as we can get to PCR-free libraries, onto the system, particularly with the lower amounts of input that are needed on the sequencer. That's really where I would like to do some experimentation, where you don't need the error correction and can really just have something that's PCR free and straightforward to do.
Okay, I guess one last question, which I think I can take this one. How does the conversion work, and do you expect that it would result in less accuracy? So basically, the conversion is just a quick, I think Stephanie mentioned this, 5 cycles of PCR. So you have primers that are complementary to the P5, P7 adapters, and then have an overhang that is the onso adapter sequence. And so you just do a quick 5 cycles of PCR to essentially add that on to the outside of your insert. and then it can be clustered and sequenced on ONSO. And I think actually Stephanie showed some data with, so I had shown some data from PCR-free HD002, and then Stephanie had shown some data from the converted libraries. And really for both of those, we were seeing that same effect of, for the majority of the read, the data is above actually Q50. We spent a lot of time optimizing the polymerase that we use for the conversion reaction. And so we are pretty confident that it won't reduce it significantly. I tend to think of converted libraries as sort of being a lower bound for the performance that we could expect, just because it does use those additional 5 cycles of PCR. But generally what we are seeing is that we're seeing a really similar level of quality across. Okay, I think that is all that we have. Oh wait, we got another one. How does your design address the challenges of accurately and efficiently sequencing through regions with high repeat sequences or structural variants? I'm not sure if that's directed to Stephanie or to Giannis. Certainly Giannis presented a lot on repetitive sequences with microsatellites. For structural variants, again, this is short read sequencing, so PacBio is fortunate to be the only company that has really accurate long and short read sequencing. And so for things like structural variants, depending on the length of the variant and what your input material is, it may be that long reads would be beneficial there, but particularly for these homopolymer regions, I think we're really seeing the advantage as Giannis showed. Let's see. You shared some studies about machine learning experiments in regards to ovarian cancer. Do you have a paper to share, or are there other studies underway for tuberculosis and other forms of cancer?
Yeah, for us, we have a paper that hopefully will be published soon, so that because it's a very big project that took a very long time. But yes, so ovarian cancer is much more tricky in our experience. So we are finishing the endometrial cancer at this point. Ovarian cancer is much trickier to detect, but it is detectable, but not at the high, we have very high accuracy and sensitivity for the endometrial cancer. Ovarian is the most challenging, and we want to solve it by combining many different assays together to increase the sensitivity in terms of early detection, what we're talking about.
Awesome. Okay, we are very over time, so very much appreciate all of you guys sticking around to the end of this. Again, we did record this, and so we will send out a link to the recording in the next week or so. And again, if you wouldn't mind sticking around, I know that we are again over time, but if you don't mind sticking around for that 2-minute survey, we would very much appreciate it. So thank you so much to our speakers. We are super excited to have you here with us, and thanks so much everybody for attending.
Thank you.
Thank you.
Thank you.